Hello, everyone. I think that this is working and that we have started. So I wanted to welcome everyone. Thank you for joining Young Chemist Fighting Coronavirus, a global conversation. I would like to start on by thank you everyone that has been involved, the International Young Chemist Network, IUPAC, UChemist, and all the people that have contributed to the organization of this event. I just reminded for all the attendees currently joining or that want to do any question, feel free to send us your question on the chat below on your GoToWebinar chat. And I would like now to hand over to Professor Javier Garcia-Martinez, the President-elect of the IUPAC, who will say also a few words of welcome.
So hello, everybody. Thank you for joining us. Let me, on behalf of the International Union of Pure and Applied Chemistry, welcome all of you to this special seminar. And I've been asked to say a few words about the role of chemistry in fighting the COVID-19. Let me start from the beginning. Okay. So, a few words just to thank everybody who has been involved, especially the European Young Chemist Network and the International Young Chemist Network. I think this shows a lot of leadership from the young chemists that we have. And again, on behalf of IUPAC, I want to congratulate all of you for your leadership and also to the speakers that we are going to be learning from today. I especially like the fact that this is a global conversation. I like the fact that we have people from the United States, from Nigeria, from Brazil, and from Germany. I think this is a global conversation because one of my key messages today is that this pandemic is, of course, a global tragedy. Pretty much every country has been or will be affected by the COVID-19. And also, my second message is that this pandemic is spreading incredibly fast. So we are not well prepared, but science and chemistry in particular has been responding incredibly fast to this emergency. The first cases were reported at the end of 2019, and already in January 9, we already had the first genome sequence of this virus, the SARS-CoV-2 virus. So we know now the genetic code of this virus, and with that we have better tools to understand how to fight this crisis. With the sequence of the genome of the coronavirus, we have been able to compare the new version of this coronavirus, the human SARS-CoV-2, with the previous version that we know very well. This is the SARS from the outbreak in 2003, but also from other coronavirus which are in different animals, bats and pangolin mainly. So in this very nice paper in Nature Medicine, we already know that this coronavirus is indeed coming from animals and not from any labs or any accidents. So this is really coming from animals, like other outbreaks that we have experienced in the last few years. So science responded very quickly. In just in a few weeks, we already knew what kind of coronavirus we were fighting. But there are also important features in this coronavirus that scientists in general and chemists in particular have helped us to better understand. And that one of these key features is this protein S, which is key from the for the coronavirus to inject its genome, the genetic code of the virus inside the cells. In this fantastic study, again, just a few weeks after the outbreak in China, a global effort of different teams from all around the world have already identified the potential binding sites between the human proteins and the coronavirus proteins. And thanks to this mapping, scientists have already identified 69 potential treatments for the COVID-19. In this paper, the authors not only suggest these 69 different molecules of existing treatments we have for other illnesses, but also relate the different ways of how this coronavirus can be interacting with human proteins. So a very detailed understanding of the interaction between human and coronavirus proteins, which are going to help us design better treatments against the coronavirus. Because of that, the World Health Organization has already identified 4 potential treatments, and I highlight here these molecules, or the main molecules involved in this global study, that are targeting different steps in the development of the virus. So solidarity is a very important effort that I want to highlight during these first minutes of my presentation, because this again shows how the scientific community has responded very quickly to this pandemic, and we are already exploring 4 very promising treatments, and I'm sure that during this webinar we are going to be learning more about these and other potential treatments. Another example of how chemistry is contributing to solve this global pandemic is this amazing study published just a few weeks ago on ACL Central Science, in which thanks to computers, very advanced computers, now we have a simulation of the virus with atomic resolution. So it's a 160 million atom simulation that help us to better understand how the virus looks like, and is a very important step into designing and having new treatments to fight the coronavirus. The level of detail in the understanding that now we have on the surface of the coronavirus, these key proteins, how it infects the human cells, is unprecedented. I just want to once again emphasize how amazing is that we have this kind of understanding so quickly, just in a few weeks after the first case is reported in China. But chemistry has contributed also, I would say, in more simple but very efficient ways to fight coronavirus, and also other microorganisms like bacteria or other viruses in general. And it's so important that we follow the recommendations of our authorities. And a very simple one, like just washing our hands, following very clear indications, with soap, it has a very positive effect in avoiding the spread of the illness. Why chemistry and soap in particular are so efficient? Let me spend just a minute explaining why soap is so efficient, because this is just wonderful. So in the membrane of the coronavirus, there are these lipids, this bilayer that is protecting the genomic code of the coronavirus. These worms that you see here actually are surfactant molecules present in soap. These surfactant molecules can penetrate in these membranes and actually dissolve the membrane, destroying the protection of the coronavirus. Also, small amounts of water can get inside the virus. That really helps to fight the virus. And as I said before, now the protection that the coronavirus has can be removed with these surfactants, which are present in soap, and also removing these proteins that they are really the way that the virus has to infect other cells. So very simple chemical has a very important role in fighting coronavirus. This is probably one of the most striking graphs that we have in our human history, if you wish, and probably the best way, the most clear way, the most visual way to explain how chemistry has contributed to the well-being of humans. For many years, the life expectancy of humans was below 40 years. Pretty much for all our history, most people die before 40 years of age. And something amazing happened, as you can recognize, at 19th century, during the Industrial Revolution, where chemistry and other sciences start to contribute with sanitation, hygiene, but also vaccination, rapid diagnostics, drugs. And because of that, now in a few years, we have doubled our life expectancy. So chemistry is key to fight COVID-19, coronaviruses in general, infections also caused by bacteria, and in general, to allow humans to live longer, healthier lives. And in a very clear representation, and this will be my last slide, in this very clear representation, when we plot a life expectancy versus GDP, so we have a better way to normalize different countries, we see that those with better sanitation, in pale blue, have a higher life expectancy than those which actually have worse sanitation. So sanitation is key to improve our life expectancy, and as you all know, soap, bleach, chlorine, of course, in chlorination of water, to have safe, clean water to drink, has a very important role in our health. With that, once again, the purpose of my introduction was merely to describe in a few minutes how chemistry in general and science in particular, and science in general has a key role in fighting COVID-19, how quickly the community is responding, and again to thank the organizer for your leadership. I'm very proud of everybody who has been contributing to this webinar, and also to the speakers, because I do think that we are going to have a true global conversation today. So thank you very much, and if you have questions, we may have a dialogue after the presentations. Thank you.
So thank you, Javier. I think this is a really nice presentation for as an introduction, and thank you so much for helping with the dissemination and the organization of this whole meeting. So now I would like to lead on with the first speakers. They will be Dr. Angela Zhou and Dr. Cynthia Liu. They are both coming from CAS, the Chemical Abstract Service. They will give us some news on how chemistry is fighting COVID-19 with some insights and strategy for drug development and vaccine development. Angela Zhou is an information chemist at CAS, and she earned her PhD in molecular pharmacology and toxicology from the University of Southern California, and she has further trained as a cell biologist at Johns Hopkins School of Medicine. We also have Dr. Cynthia Liu, and she's the leading expert on the article that Professor Javier Garcia was speaking earlier on ACS Central Science, and is the currently manager of scientific information at CAS, and they will both be able to now give us these insights and strategy for the COVID-19. Please, whenever you can, get away with your presentation.
Okay, I'm ready. I just unmuted myself, and can I get the share button? Okay, I see it. Share my screen. Okay, can everybody see the slides?
Yes.
Okay, great. All right, thank you so much for having us here to share our insights and the strategies for drug and vaccine development on this COVID-19 situation. My name is Angela Zhou, and I'm here joined by my colleague, Dr. Cynthia Liu. We're both information scientists from American Chemical Society. We're a division of we're a division of American Chemical Society. The CAS, we do chemical abstract services. Now, as we all know that the COVID-19 has become a pandemic situation, and we are seeing the confirmed cases rising every day. Each day, thousands of people are dying from this disease. The disease has impacted a lot of people's life, and our global economy has been impacted significantly. Luckily, we are seeing this ongoing worldwide effort in the scientific community. We try really hard, try to identify the effective drugs and develop vaccines as soon as possible to fight this disease. So from this map, we can see the disease is widespread around the globe, and it has spread to 6 continents. Since the crisis of this COVID-19 situation, many scientists at CAS have been worked together and published a report on this COVID-19 regarding possible drug candidates and vaccine strategies in ACS Central Science Journal. If you're interested in more detail of our work, you're more than welcome to download our paper from the link that I provided here. Besides this work, many scientists at CAS are also working hard to get a setup database which contains nearly 50,000 of compounds with known or potential antiviral activities and their related information to support this COVID-19 research. This data set is available for download to the public. You can download from the link that I provided here. Now, what exactly is CAS? What CAS can do and what CAS scientists are doing these days to fight against this terrible disease? We have hundreds of scientists who are analyzing and connecting the world's scientific data around the globe. We work with more than 50,000 of scientific journals, and we work with more than 60 patent offices around the world. We have accumulated more than 150 million of substances, which including small molecules as well as biological sequences. We have scientists who altogether speak more than 50 languages around the world. So although we are named under the Chemical Society, but we cover way more than just chemistry. As you can see, we have disease information, we have cell lines, DNA, RNA sequences, and a bunch of other information such as pharmacology, toxicology, and so on. So we are eager to help in this situation. Now, having said that much about what CAS are, what this COVID-19 situation, so what is that invisible enemy that we are facing every day? Like Dr. Martinez has showed us the nice video. This is a virus just given an official name called SARS-CoV-2 virus. This virus belongs to the beta coronavirus genus. And as we can see from this cartoon image, the virus contains a single-stranded positive sense RNA genome. Because this is a positive sense RNA, the virus actually is ready, this piece of RNA is ready to be translated into viral protein once upon the virus is entered into human cells. And the virus itself is encapsulated by the viral membrane. This membrane is lipid bilayer. As Dr. Martinez mentioned, it can be disrupted by soap, so washing hands is really important. The membrane is embedded by many envelope protein as well as covered by this famous S protein here, colored with red color. This S protein, it is because this S protein, it has very high binding affinity to a human cell's receptor called ACE2. Because of their high binding affinity, allows these viruses high contingency, and upon this binding, the virus is able to enter into human cells to do the viral infection process. Now, since the outbreak of COVID-19, scientists at CAS has been paying a close attention to what's going on the field. We realized that there's an explosive amount of number in the journal articles related to COVID-19 being published at the end of January and the beginning of February. The number continued to increase these days, which reflects a very good scientific community's effort that scientists are trying to study this virus mechanism and try to figure out the best solution for this situation. Now, looking back in our existing database, we try to see what we can learn from our previous experience to fight these SARS-CoV-2 viruses. We realized that there are 2 episodes very similar to today's situation, which are caused by SARS-CoV virus and MERS-CoV virus. Together with SARS-CoV-2 virus, these 3 viruses, they all belong to beta coronavirus genus. And there are some similarities we can learn from our existing experience. Hopefully we can borrow this experience to fight for these novel coronaviruses. For example, both SARS-CoV-2 and the SARS-CoV virus share the same human viral receptor ACE2 protein. And more interestingly, both SARS-CoV-2 and the SARS-CoV has the S protein with very similar protein sequences and a receptor binding domain. And because all of these viruses are coronaviruses, they have these major viral proteases. These major viral proteases are required for viruses replication in the cells. And also, again, because these are RNA viruses, all these viruses require a RNA polymerase called RNA-dependent RNA polymerase. We're hoping to see if there are any existing small molecules which has been studied, showed some inhibitory effect to this RDRP protein that we may be able to borrow to use for the SARS-CoV-2 situation. And more interestingly, scientists also find that there's a human protease is actually secreted from human cells. It is called TMPRSS2. These proteases is required both by MERS-CoV and the SARS-CoV-2 virus to process their S protein, so the S protein can bind to ACE2 receptor on the cells. So having this information in our mind, scientists at CAS try to look in our existing database to see if there's any potential drug candidates and the vaccine strategies with SARS and MERS that we can borrow for today's situation. In our paper, we covered 3 major areas. The first one, we talk about small molecule drugs and the drug candidates. We also covered biologics. Biologics are a group of therapeutic reagents. They're actually produced from biological systems, such as protein like antibodies, cytokines, or DNA, RNA, large molecules. And we also talk about vaccine strategies, like what type of vaccines and what type of technologies are involved in vaccine designs. But for today's talk, I will mainly focus on the small molecule drugs and the drug candidates. We will talk about some of the key proteins and their roles during SARS-CoV-2 infections. Hopefully, these key proteins can be used as drug targets for small molecule drug development. And then we will use these key proteins as baits to search for small molecules in the research and the development for SARS and MERS research database. And finally, we also wanted to bring up some of the existing drugs and for their potentials to be repurposed for COVID-19 situation, because drug repurposing can really save us some time to find some effective drugs to treat these diseases. So as we have mentioned before, this novel coronavirus SARS-CoV-2 has several similarities when we compare with SARS-CoV and MERS-CoV. First traits is that they all have these corona protease, major proteases. These proteases are required for the virus to process their polyprotein into functional smaller protein units. So the virus can use this protein to make RNA replications and also to assemble their viruses. There are existing drugs that inhibit viral protease. Lopinavir and ritonavir are drug combination for HIV viruses, actually. So these drug combinations are currently under the clinical trial to see if they can inhibit the coronavirus's protease. And we've also mentioned that this key protein here, the RDRP RNA polymerase, this enzyme is responsible for the virus RNA polymerization. And there is a drug candidate called remdesivir, which is originally developed for Ebola virus. But this virus, during the developing process, it does show some inhibitory effect against SARS-CoV and MERS-CoV. This drug is also in clinical trial to see if it has any benefits to the patient with COVID-19. Now, of course, There are many other proteins, play a key role during the viral infection, and there are possible existing drugs or drug candidates may work against these key proteins. And I don't have much time to go over each one of them. And you are more than welcome to look at these targets in our paper. I'm sorry, I have to mute my.
Well,
I'm so sorry. Accidentally the phone rang. All right. So looking at our existing data, we also try to find if there are any patterns related to SARS and MERS situations. We did discover there are more than thousands of patents related to SARS and MERS, and many of those are actually related to therapeutic reagents. And we also find there are many patents related to vaccine development strategies. And we are hoping to learn from this existing experience to get some information for this SARS-CoV-2 situation. Again, like I said, there are many key proteins, function critically during this SARS-CoV-2 infection process. We used these targets, key proteins, to search for existing drug candidates, see if there are any possible hits that we can use as raw material to start the scientific research. So indeed, we find more than hundreds of patents related to these key proteins and related to SARS and MERS situation. And in each of these patents, it includes several drug candidates, small molecule drug candidates, and we're hoping to see the inhibitory effect to each of these target proteins here. And finally, I would also like to bring up this drug repurposing table because drug repurposing can really save us some time to go against this COVID-19 situation. I want to particularly talk about this baricitinib drug. This is a drug for rheumatoid arthritis because it has inhibitory effect towards the inflammation process. But this drug was identified by AI. The AI program discovered that this drug may have some inhibitory effect for the virus to enter into the cells. So with these double effects of baricitinib, of inhibitory, inflammatory, as well as inhibiting the virus entrance to the cells, this drug may be very beneficial to COVID-19 patients. And we are seeing this drug is currently in the clinical trial. And there are several other drugs such as chloroquine that Dr. Martinez also mentioned as well. This drug actually can block the glycosylation of ACE2 receptor on the human cells. Now, without the proper glycosylation on the ACE2 receptor, hopefully the virus can no longer recognize this receptor. And this chloroquine can actually also disrupt the cellular pH level. We know that a lot of viruses, in order for viruses protein to work, it really depends on the acidic pH environment in the cell. So hopefully with these double effects of chloroquine, it can inhibit the viral infection in the COVID-19 patients as well. Now, at last, I also wanted to bring your attention to the vaccines that we identified that are related to SARS and the MERS situation. And there are a wide range of vaccines here across different types of technologies. I want to bring your special attention to this mRNA vaccine technology. Although we do not see many of the patent numbers associated with this technology, but this is a very new technology and supposed to be safer than most of the traditional vaccine technologies. The one that was designed and currently in a clinical trial, it is based on the mRNA vaccine technology. From here, I would like to conclude my talk. We did talk about the key proteins that play critical function during the viral infection process, and we are hoping to see that some of these key proteins can work as drug targets for small molecule drug candidates. We also used these key proteins as baits to search for small molecule drug candidates in research and development, particularly associated with SARS and MERS viruses. and hopefully we can use these, small molecules to for the COVID-19 situation. And we also talk about some of the existing drugs, how they may be potentially repurposed to go fight this COVID-19 disease, because these can really save some time since the whole world is in urgent need for effective medicines. Finally, we also talk about the vaccine technologies. And at the last, I want to bring your attention that what CES is doing and what CES scientists are trying really hard to support the COVID-19 research globally. We do provide custom curated data sets of drug candidates and the targets. We also have patent landscape analysis related to scientific technologies. We have a wide range of scientific expertise which can provide consultation and services per your request on your research topic. So from here, I would like to thank you for your attention, and we are welcome for questions in the panelist discussion.
Thank you, Angela. This is a really nice presentation. I have gathered some of the questions from the public, but if we also have some of the staff or of the other presenters questions, we have a little bit of one or 2 minutes. Otherwise, I will go ahead first with the question of the public.
Okay.
I have one question here from Katerina Arvaniti, that they are asking about the size of the virus, because apparently this virus is completely different from previous generations. I mean, for the public in general, they find it that viruses are, or in the imagination, are really small. This one is, from my understanding, relatively big, right?
Yes, yes. Can you hear me? Yeah, this virus is relatively big, yes.
Oh, good. But we don't know the size of the virus, right? Or I think that even in Javier's talk, they were mentioning the already known structures of the virus.
To be honest, I don't know the exact size of the virus, but we know that coronavirus, they are relatively large, virus particles compared to most of other viruses. So in terms of the structure-wise, they are relatively sophisticated if we compare with most of other viruses. So they may have better mechanism to to go against drugs if we wanted to fight against these viruses. So they may have a more sophisticated mechanism to fight with some of the drugs we want to use.
There was also a question from Janela Lasik that's regarding the hygiene recommendation that has been around. It says that the recommended time has been for washing your hand is around 20 seconds minimum, and she's asking about, is it truly enough time for the shock molecules to damage the structure of the virus membrane, or how much time will it be necessary for this to happen?
I can take that one. In terms of the size of this coronavirus, is around 120 nanometers in diameter. So it's a little bit larger compared with other viruses, which with the capsid is a little bit smaller, but definitely in the nanometer range. So yeah, big for a virus, but not huge. In terms of the washing your hands, yeah, like with any other grease, any other oily substance, it is important more than time to make sure that you wash your hands properly. And if you have the feeling that your 20 seconds is not enough to destroy that membrane, maybe a little bit more. I'm been hearing a little bit longer times, but I think the authorities are worried that if they say something like a minute, people will just not follow. Actually, it's a very efficient way of destroying the membrane. Yes, and in the same way we can remove grease from our hands just in a few seconds. In the same way, the surfactant molecules can penetrate the lipid membrane and destroy the virus quite effectively. Again, the most important thing is to do it properly. You have seen the videos, I don't need to repeat, but yes, just a few seconds can make a difference. Exactly.
Yeah, there has been some people that they have been speaking about the role of how the molecules of water could destroy the bifacial layer of fat. But I think that we can continue and we can leave. We have quite a few questions from the public as well, but we can continue with the conversation and leave it for the after Q&A presentation. So thank you so much, Angela, and also Cynthia, for this presentation and for your work. I would like now to introduce Dr. Emmanuel Balogun. He's connecting from Nigeria. So he's going to speak us about the prevention and control of coronavirus 19 from the perspective of the chemist. His research interests are based on diverse areas on the biochemical sciences, with his special interest in the development of cures against African endemic diseases. Emmanuel is the recipient of the Gold Medal Prize Award for Science from the Nigerian Academy of Science, which is the highest honor given by the Academy. And he also holds the Fair Fellowship from the Royal Society in the UK and other African chemical societies. So, Emmanuel, whenever you are ready. Oops. Emmanuel, we cannot hear you.
Can you see my screen?
Yes, we can now hear you and see you. So please go ahead whenever you want.
All right, thank you. Okay, yeah, good to meet you guys, meet you all. Thank you for this opportunity to discuss with colleagues here on this forum. So my title, as you have seen, is Prevention and Control of COVID-19 from Perspective of Chemists. As Professor Martinez mentioned, chemists have played a significant role in daily living of humans, and we have really contributed towards quality of life and longevity, good health, and everything that you can think about in terms of promoting quality of living. So this is what we're talking about, the pandemic, which is the major issue, and a number of you have seen these maps, the regions that are much more heavily affected, and some other regions are much less affected. I will take this opportunity to mention here that because I have received a number of questions from colleagues all over the world that how comes Africans are not so much affected. But I think Africans, we do not have any form of immunity. We are all the same. We can be affected. Actually, there are lots of infection in the region of thousands, even within this week in Africa. Although I must say that perhaps maybe it's a milder form that we are experiencing at the moment. We don't know what will happen in a short while. Again, just riding on the introduction by earlier presenters, COVID-19, what is it? It's a pandemic which surfaced in the globe by the end of 2019, and it has been with us. Hopefully, we will be able to surmount this current challenge. It's caused by a newly identified coronavirus, which is called SARS-CoV-2, and this is one of the 7 actually coronaviruses that can infect humans. Although 3 of them, the SARS-CoV, and then MERS, and then the present SARS-CoV-2, are the ones that cause severe diseases in humans. The other 4 present mild symptoms. I will not go into details of the structure of the particle itself because already the previous speakers have done a good justification about the introduction. But just to mention again is the fact that this the spike protein is very important. It is also a positive single-stranded RNA virus, meaning that the genome is single-stranded, just like it has been mentioned. So usually before beginning my lectures, I usually show this slide and to define life. And I always define life to my students and colleagues as a concerted, well-synchronized chemical reaction that proceed. That's just simply the definition of life. It's highly regulated chemical reaction that proceed in living organisms. And usually these reactions proceed in a highly regulated manner, and that's why we can have good health. And when there is dysregulation of these pathways or these chemical reactions, it can lead to diseases, it can lead to death. So now looking at the SARS-CoV-2, it affects the lung cells, and I will show in just a short while how it gets into the cell, although it has been mentioned previously. And then because it affects the lung, the organ, the lung, and the cells of the lung. So that meaning that all reactions that are peculiar to lungs will be affected. And of course, as we know that one of these things is the oxygenation of the entire system. So if this is affected, then the entire system will be compromised, and it will lead to what we have seen as one of the symptoms, pneumonia of SARS-CoV-2, and eventually death. It has been said that the origin of this virus is bats, and then it gets to an intermediate. Now it's not known, but one of the major suspicion is that a pangolin, and then which gets in contact with human, and then we can have a human-to-human transmission, which is majorly in the form of droplets when an infected person sneezes or coughs, and this is why this has informed most of the interventions that we have all been talking about. Now, looking at the virus, how does it get into the cell? Because the virus, it's a particle, it's not a living thing. But strangely, it has some machinery that is able to anchor it to the structure of the cell. Just like Angela mentioned, it's the spike protein, and this spike protein recognizes a receptor on the cell surface, which is the ACE2. And then when this binding occurs, there can be two mechanisms of entry, which is receptor-mediated endocytosis or just membrane fusion. And when this occurs, then the viral genome can just be released into the cell. And once in the cell, it's because the positive strand, RNA, so some part of that can be processed directly into an mRNA for some machineries that helps in the viral replication, and also in transcription, and also packaging of the viral genome into the capsid that is synthesized using the host procedure. So we look at this, the genome. It's the size of the genome. It's not so big. It's about 30 kilobase pair, meaning 30,000 base pair in length. And this, there are some key products that can be produced from this gene, from the genome. You have some proteases, like Angela mentioned, and you have some non-capsid proteins that can also be coded for. So the binding of the spike, like I mentioned, the ligand is the spike protein. Actually, it's not the entire spike protein. It's just a particular region of in the spike protein called the receptor binding domain. The receptor binding domain is the one that is important in recognizing the angiotensin converting enzyme 2, and then this binding can with the affinity of the binding can mediate entry into the cell. A number of researchers, I'm sure Professor Martinez mentioned this paper in Nature in his presentation. I'm also using this. They try to compare the SARS-CoV and the SARS viral RBP domain, and then they found that there were some mutations that occur in the SARS-CoV-2 spike receptor binding domain that probably helps in the tight binding of the receptor to the ligand for the entry by the virus. So here I have highlighted some. I don't know if you can see my cursor. There are some regions here where you have some mutations. Mutation means some change in the nucleotide sequence, leading to a change in codon, which means leading to a change in amino acid. Here you have one of the changes we have seen here. The asparagine, the glutamine residues of the RBP domain, and this helps in the tight binding of the ligand to the receptor. And this has been attributable to the high transmission rate. Transmission rate means how many people can a person be able to transmit the virus to in the course of the infection. So this tight binding, as a result of this mutation, have probably helped in the infectivity of the virus that we are talking about. So now, looking at the infectivity and the spread of COVID-2, if you look at the slide here, on the left side, if you do not have any intervention, an infected person can transmit to at least 3 people, between 2 to 4 people. But let's look at this scenario where an infected person can transmit to 3 people. So, and then each of the 3 will go ahead to transmit to another 3, a set of 3 people again, and then this continues. And then before you know it, you have this expansion of infection among the population. But if you have an intervention in which we can have, maybe the intervention can be testing and isolation, as we have seen here. If an infected person is tested and recognized, can be isolated, it means subsequently he will not be able to transmit the infection. If you don't have this kind of intervention, then the person will go ahead to proceed to transmit it to other people. We can also have some personal preventive measures, which will bring me to one of the points that Professor Martinez mentioned. And these personal preventive measures can be, I categorize them into physical and chemical. Physical means, an example is what has been advocated by WHO, the social distancing. That is at least 1 to 2 meters in between 2 people in the particular. So it helps because the virus is spread through aerosol, and we know that they do not remain in the air for long. They obey gravity and fall, and therefore it cannot proceed to the next person beyond 1 to 2 meters. The other one is the use of mask, and I don't know, maybe I don't want to overemphasize this, but I will use this opportunity to advise that if possible, people should use masks. This is a form of physical barrier, but studies have shown that this is also very effective. And of course, as chemists, we have seen that washing with soap or detergent for 20 seconds is very effective. And importantly, also is the use of hand sanitizers as frequent as possible. The hand sanitizers contain ethanol. Ethanol is a solvent. It will dissolve the envelope of the virus or an infected cell, and therefore it will inactivate the particle from being transmitted. Now, again, one of the role, apart from prevention and chemotherapy, one of the role of chemists in this pandemic is in the design of testing kits. I think the most widely used testing kit is the use of qPCR. This PCR means polymerase chain reaction. It's a way in which a strand of a piece of DNA material or a nucleotide material can be amplified to number of copies up to the extent that it can be visualized or quantified using the machine. So in this testing kit, I think what the consensus is that there should be at least three targets. I think one of the targets is ORF1, which is one of the regions in the genome that targets the non-nucleocapsid region. And then you have the N protein, which is around the nucleus, and then you have the S, that is the spike protein. So when you have one of these being identified, it means it's false positive. And then if you have two, if you have one, it's inconclusive. If you have two, then there is very high chance that the person from which the sample is collected is infected. And this machine can be used to quantify the amount of the virus in a particular sample. Angela mentioned this, the use of chloroquine, and so I saw this paper very recently, which was published, and where they have tried a combination of hydroxychloroquine and azithromycin. As we know, hydroxychloroquine is an analog of chloroquine, chemical analog of chloroquine, although it has some chemical features that makes it much better in terms of toxicity, that we usually know associated with chloroquine. So hydroxychloroquine seems to be very potent, but the effect is synergistic when given in combination with azithromycin. And azithromycin is one of the drugs that is used even in the treatment of pneumonia. So I guess that informed the investigators to try to combine these drugs. And so, well, from their data, it looks promising. And I understand there are still lots of trials going on to be able to validate conclusively about these two drugs as a therapeutic approach against SARS-CoV-2. So just to give a wrap-up, as chemists, what can we see from all this? When we look at the pathway, the life cycle of the virus, we have been able to, Angela mentioned some of these targets, drug targets points, such as the spike protein, which has been recognized to be a very good vaccine target candidate. And I think a lot of trials are ongoing in this regard. And then you have the other proteins or enzymes of the virus that is also being targeted. She also showed us some a collection of some of the compounds that have been used to assess this. And again, of course, we have processes because the virus needs to be processed further in order to be able to gain entry into this. To gain entry into the cell. So now, in conclusion, we cannot overemphasize the fact that for now, what we have, we do not have a vaccine against this disease to prevent this disease yet. And again, also, we do not have consensus medicines for the treatment of this disease yet. So therefore, it will be very, very important and responsible for all of us to still hold on to these very simple ways of preventing the spread of this disease. If we know we are infected or we have symptoms, we isolate, or even without the symptoms, we should, when we go out in very highly endemic regions, we should use the mask. The mask serves as a barrier. It can help to reduce the rate of infectivity, and of course, using hand sanitizers and washing of our hands regularly, which basically is about improving our personal hygiene, will go a long way to help. And this disease really brings to my mind, I didn't emphasize that, I didn't know that, it didn't come very strongly to me, that there is really very high interconnectedness between we humans and animals, and also even we humans among ourselves. So therefore, it is very important to keep this at the back of our mind to be able to help ourselves and our family. Thank you. I will entertain any question.
Thank you, Emmanuel. Thank you so much for this interesting presentation. I have some questions from the public, but I would like first to open the questions if any of the other panelists have any other questions, or I can more or less give a couple of them that I can find it more interest from the chat that we have from the people asking, and then maybe we can intervene as the time Professor Javier Garcia did when there was a question. Okay. I have you were mentioning on the spread of the virus regarding the respiratory system, but there is some common misconception, and I don't know if you can explain it. Some people are saying that can the virus enter your body via your eyes if they are not covered?
Yeah, the respiratory system usually is a target. We know that it goes to the pulmonary system, meaning it goes to the lung, and the entry point to the lung, basically, of course, majorly is through the mucous membrane of the nose and also the mouth. But of course, we also need to be careful about our eyes, because sometimes there is a connection. Not sometimes, there is a connection between the eye and the trachea and the respiratory tract. That's why sometimes, I don't know if any of you have used an eye drop. Sometimes when you use an eye drop, you feel it in your throat. So that's the connection. So that's why we should try not to contaminate our eyes, because it can just go to the respiratory system.
Okay, thank you so much. I have a lot of questions. We have a whole team of people behind that are compiling all the questions from all the public, and we are getting them there. I would like to go all over there, but we need to continue with the program. So we will go back with the questions. But I would like to continue with our panelists. So thank you so much, Manuel, again for your presentation, and we will now go on with Dr. Jadel Kratz, that is from the Drug and Neglected Disease Initiative in Brazil. He's let me show you here. He obtained his PhD in pharmaceutical sciences at the Universidade Federal de Santa Catarina in Brazil. He's a discovery manager at the Drug and Neglected Disease Initiative, overlooking scientific and operational coordination on the collaborative drug discovery platform for neglected tropical diseases in Latin America. He has held other academic positions, such as the professor assistant at the University of Vienna. So now from Jadel, whenever you want, you can go with your presentation.
Oh, good. Thanks, Antonio. Let me see if I can share my slides. Okay. There you go. Can you see my slides now?
Yes, we can see you, and you can see your slides.
Good. So I'm gonna put it on
screen mode. Yeah.
Full screen? No? Yeah.
Yeah.
There we go.
Yeah.
Well, good. Well, thanks again for the invitation, and hello everyone. It's a great opportunity actually to talk a little bit about the situation on COVID here in Brazil. I'm just going to give a very quick overview on that, and then talk a little bit from a slightly different perspective about this disease and more how it's impacting the science, how science organizes, and how scientists all around the globe are collaborating and shifting a little bit their goals and their modus operandi, let's say, to a more open science of things. And this is very much aligned with the vision the NDI has for the R&D environment around neglected tropical diseases, but this is also true for COVID. So I'm going to talk a little bit about that. I hopefully this is interesting for the audience, and I'll be happy to answer questions afterwards. So as we've been speaking, COVID is of course a huge health challenge, probably the largest in our generation, and it will have massive impacts on both the human side but also the economic consequences. We are by today approaching more or less 1 million cases with about 50,000 deaths all over the globe. So as many of us already spoken, this impacts all our communities, but also as a species around the globe, this is very, very critical. And it's no different here in Brazil, of course. By today in Brazil, we have confirmed cases around 7,000 cases and around 250 deaths in the country, mostly around old people. So it's following more or less the trends around the globe. But of course, every community, even though we're all connected, it faces particular challenges and also have particular opportunities to deal with the epidemics. And this is of course true, that in Brazil we had a timing opportunity. I mean, the epidemics arrived here later than other areas, so we had more time to prepare ourselves here. Also, Brazil is less connected than other countries, so we have less mobility of people, which also helps. The climate questions, we're still leaving the summertime going to winter now, might have helped. There are indications that climate might help the seasonality of the virus, although this might not be a critical path. And also demographics of our population in Brazil might have helped for this, let's say, lower number of cases if you compare the size of Brazil with other countries. Also, the country has early adopted isolation measures and also building hospitals in order to assist the sick people. But it's also very true that we have severe limitations regarding testing. So these numbers might be suboptimal or sub-notifications of cases. Brazil has a very long history of dependence regarding medical and pharmaceutical consumables. More than 90% of everything scientific and medical facilities consumed here are all imported. And of course, those include testing kits or personal protective equipment and active pharmaceutical ingredients to prepare drugs. So all that is very, very much important to combat these pandemics comes from abroad. So Brazil has severe limitations on there, where the demand is high and every area is competing to put their hands on those resources. Also, Brazil has challenges of its own, not only Brazil, but other countries in the region, because we have poverty around our populations, which impacts the infrastructure. So these might be specific challenges, how to contain the spread and how to provide the assistance needed in settings where resources are very much limited and people are gathering and living together in not ideal areas. And apart from that, also, the countries such as Brazil might have issues regarding co-infections. So we have a lot of tuberculosis cases in Brazil and other viral diseases, mosquito-borne diseases such as dengue. We have a massive epidemic of dengue going on in the country right now in parallel with COVID. So these might bring challenges of our own. And so although this is of course a global problem, and we must face it together, it might have specific impacts in resource limited areas such as Brazil. But as I mentioned, the topic I want to bring your attention to today is more how this pandemic might be affecting the way we collaborate. And I just saw this very nice piece of journalism in the New York Times published yesterday by Matthew Puzo and David Kirkpatrick, where they discuss how scientists are collaborating in such a large extent never seen before. Maybe something seen like this around the HIV epidemics in the 90s. But without precedent here. And I really like the quote that although we had to shut down our borders for one reason or the other in terms of isolation, the scientists have been shattering their walls and actually creating a global collaboration unlike in history. And this is very much aligned with the vision the NDI has in terms of collaborations. And this might have happened for several reasons. It might have been because all the research had to be stopped in labs and industries around the globe because of the epidemics, or it might also have been because we have to respond all together in terms of survival as species. But nevertheless, there's no easy way out. There are no shortcuts, and collaboration must be the key role when we rethink our approach for R&D in order to attend the patient's needs and also to be prepared for pandemics such as the COVID-19. And I'll take the opportunity to talk a little bit about who we are at the NDI. So the NDI stands for Drug for Neglected Disease Initiative. It's a non-for-profit organization. It was created back in 2003 with the goal to discover, develop, and deliver treatment for patients affected by neglected tropical diseases all around the globe. So we work through a network of partners around the globe. We raise funds from public and private donors, and we remove the costs and the risk from the development of new chemical entities or new treatments or new fixed dose combinations for these patients in order to deliver affordable, but also drugs that are being developed focus on the patient's need. And by doing that, actually fulfilling the gaps in the R&D in the system. And by doing that, we do that including partners all around the globe, including partners for endemic areas where capacity strengthening is very important. And by doing so, the NDI was able to deliver 9 new treatments for this disease, saving millions of lives so far. I'm not going to go deep in terms of the model, but the NDI was created back in the time to fulfill the needs of neglected patients, mostly from diseases caused by parasites and or neglected tropical diseases such as Chagas disease or filarial disease or leishmaniasis. But from the time it was evolving its portfolio and started working with other niches where the classical business model of pharma didn't work, such as pediatric HIV or antimicrobial resistance. There's a huge issue with resistance on bacteria. And of course, I mean, new emerging infectious diseases such as COVID-19 also could benefit from this alternative health-driven R&D model the NDI has. And the NDI has launched, together with several other partners from the globe, a new coalition called COVID-19 Clinical Research Coalition. It was launched this week. I'm not going to go into details. I just wanted to mention that this coalition is focusing on accelerating clinical research in resource-limited settings, mostly countries from Africa, Latin America, and Asia, in order to take into consideration the needs of the patients in these areas, because we cannot fight this pandemic by only conducting research in specific areas. We need to conduct this as a global collaboration involving all patients that are in need at this time. But as I said, I don't want to go specifically on the NDI, but I wanted to bring your specific attention to one of the, let's say, one of the pillars of the DNDI model uses in order to address the gaps in the current R&D process. And this, the feature that I think is most relevant for us today is actually I don't know if you can see my mouse here, but it's the collaborative, the open and transparent aspects of the R&D. As I mentioned, COVID-19 already brought people together, scientists working together with governmental agencies, regulatory agencies, patients, communities, pharma, from different spheres from all over the globe. And this is very much how the NDI works since its inception. It has a huge network of partners around the globe, from low and middle income countries, but also high income countries, all working together under the same goal. And this is very much the spirit COVID has brought many of the current initiatives. So this is one of the key aspects that we would like to highlight, and this should be the, let's say, the normal practice around science. Science should have no borders and be a true collaborative approach in order to have most of its impact, actually. And also another aspect of the model is to be, the NDI has been a long-term advocate for open science initiatives, and open science is a large concept. It can mean a lot of things, but it's mostly related to sharing scientific information as fast as you can in good quality and also free of charge. That this public or open access to scientific data is available for all the community and could make impact and connect the scientific world, let's say, with the community and all other spheres as fast as it can. It can be more efficient by sharing the resources, avoiding duplication. It can be more reliable because it's better verifiable by the peers, and it can quickly and better respond to the society's needs. And if you see what's going on through the COVID pandemic, it's very, very clear. You can see all the paywalls from the publishers being lifted. I mean, all the publications related to COVID-19, or at least most of the publishers, or even journals and scientific journals, releasing these publications for free, which is very good. So the community and the decision makers are assessing the latest scientific data as fast as it comes out. Imagine if this was the current practice for all diseases. This would be fantastic. Also, the shift in terms of practice for publishing regarding using of preprints. Of course, these preprints are these fast publications that haven't gone through peer review yet. They have advantages and disadvantages, but their fast publication of urgent and very much needed data is very important to support the quick decision making that we need. And we must reward researchers that use these type of resources, preprints such as medRxiv, ChemArchives, and others. Also, open data repositories where the community can access resources from data. Angela already mentioned the one from CAS, but there are others such as Campbell, such as the PDB, where you can find in vitro, in vivo data, also information about protein structures, ligands, crystallography data, or platforms such as the NextStrain, where all the sequence from COVID-19 for SARS-CoV-2 have been released and organized in phylogenetics trees in order to establish the origin of the infections. This was just amazing work done by the participants. It was done fully in the open and open for everyone. If you think about just in a matter of perspective, in 2003, when the SARS epidemic came out, it took 20 months from the genome sequencing until the first tests of the vaccines using this information was done in humans. If you look at the COVID, the sequence from the genome was made available in January, and only 69 days after, we already have the first clinical trial with the mRNA vaccine from Moderna and NIH in humans. This is just unbelievable. It's actually amazing work done in the open, and it proves the concept. But we don't have to think only about data when we talk about open science initiatives. We can also think about tools, to make tools available. And one nice example that the NDI has been involved together with Medicines for Malaria Venture, other non-for-profit organization working in malaria, is the Pandemic Response Box. The Pandemic Response Box is actually a box of compounds. This was organized back in 2018 and launched in January 2019, when we didn't have an idea that COVID will be around yet. But it's a collection of non-antibacterials, antifungals, and antiviral compounds, 400 of them. These compounds, they are selected based on chemical diversity, but also on target, pathogen, and also mechanism of action. And these compounds, they are free for access. They are first in line available for screening around. So if a lab has a screening set up for any pathogen around, and wishes to screen these compounds, you can apply for it, and MMV and the NDI will ship these compounds to the lab. And the only commitment the group has to do is to make the data from this screening available for the community. So this is one type of initiative that might help. This was thought actually to be addressing future pandemics, and this box has now been shipped to several labs that have SARS-CoV-2 in vitro assays and are helping to bring new leads and new ideas into new chemical entities that might help to develop drugs for the COVID-19. There are other initiatives. I cannot cover all of them, but I thought I would bring some that I thought were really, really interesting. One of these is the COVID moonshot. It's a project involving several groups. You can find a lot of information online, but the idea is to get chemists or chemist students all around the globe designing inhibitors for the SARS-CoV-2 main protease. We already see my colleagues talking about some of the targets in this disease. The idea here is to develop covalent warheads, connect them to no covalent compounds for a probe discovery project using concepts that are already published. You can find more information on that. This is pretty cool. Some of the designs are already, the first 2 rounds of design has already been completed. These compounds will then be synthesized, shipped to Diamond in Oxford for testing against the main protease of SARS-CoV-2. Hopefully we'll bring new leads for drug discovery projects as well. Others are projects such as Folding at Home, where you can work to help determine protein structure through computational chemistry and biology. You can donate power from your mobile, from your computer, but you can also participate in this project. It's quite interesting. And there are several other initiatives around. I cannot name them all. Please go around. There's plenty of open science initiatives going out to help progress science for COVID-19. And just to finalize my talk, just one final reminder. I'm going to steal the words from Carolyn, chemistry professor at Stanford, on Twitter this week. She used the words, so when we are finally no longer worried about COVID, which I hope will be soon enough, unfortunately we will still have a lot of other diseases that affect people around the globe. We will have Chagas disease, leishmaniasis, malaria, tuberculosis, dengue. There are millions of people affected by these diseases that might not be easy to tackle using the conventional drug discovery model. So let's not forget about the importance of the collaboration and the openness in the drug development. These are key to actually fulfill the patient's need, and also for us to be, as a community, to be prepared for the future pandemics. And I would like to thank you, everyone, and I'm happy to answer questions now or afterwards.
Thank you, Jadel. I think that you have made a really interesting point. If someone else was doubtful of the importance of open science in this time, it has been more than clear that it is the way to go. We have a few questions, but I want to continue with the program. So just until we have a few questions that are convenient for all of the panelists, I will continue maybe with Julia from connecting from Germany now. It's Julia Kunflind. She's a PhD student at the Technical University of Munich, working in the field of analytical chemistry, and she's going to talk about the hackathons that had been initiated by the German government and the resulting projects by the Pandemic Important Research Allocation Tool, or PIRAT, which we are really hoping that the topic doesn't have anything to do with PIRAT. So sorry for the joke. I need to make it. So Julia, whenever you want, is your presentation. You can go.
So can you see my screen? Does it work?
Yes.
Perfect. So hello everyone, and first thanks a lot for the opportunity to speak here, and thank you for the very interesting presentations we heard so far. I will not really talk about chemistry, so sorry for that in advance. But what I would like to do is to show you how distance, as we have to keep it at the moment, does not necessarily keep people from uniting and working together to create great things. What I'm presenting you is a German approach in fighting corona, not because I think we are doing especially well, but because this approach showed me personally how much we can achieve if we just work together, no matter our professional background or any geographical boundaries or curfews. So I'm going to talk to you about how the German government asked 82 million people for help and how 43,000 of them actually helped. You surely all know the situation in your home countries very well. And although the number of COVID-19 cases and deaths may differ notably from what we have here in Germany, the challenges emerging from corona most probably are similar. Here in Germany, we currently have 73,000 people infected with corona, and sadly, over 800 people already died. The first case was registered more than 2 months ago, and for the last 2 weeks, we have had a more or less strict curfew that will continue for various weeks. And the main challenges we have to face are the testing capacities that are way too low at the moment, with many suspected cases that cannot be tested and people having to wait for days or weeks until they get a test result. The worldwide ongoing research for vaccines and drugs against corona that has to go on as fast and as fruitful as possible. The shortages in consumables that already led to very creative solutions in many countries. And additionally, challenges in digitalization, as schools and universities will have to start teaching online, and strategies to do so will have to be developed soon. Most of you probably already know various initiatives, be they international or from your country. Actually, we just heard about some very interesting initiatives. And some examples I find very interesting are, for example, Folding at Home from the US, which we heard about just before, or Crowdfight COVID-19, where scientists can ask for help in very specialized tasks and often get the help they need from somewhere in the world. And something very easy, the sewing of masks from people who just are able to sew things to at least try to prevent people from infection. And what the German government tried was something that has emerged as a powerful tool to bring people together over the last years. They organized a hackathon as an approach to generate as many initiatives as possible in a very limited time frame of 48 hours. But in contrast to many other hackathons, the clear aim was to develop projects that can be applied as soon as possible to help against corona. So this is how the Wir versus Virus hackathon was born. Wir versus Virus means we versus virus, as most probably wasn't too hard to guess. It was initiated by the German government and took place from the 20th to the 22nd of March under the patronage of the head of the Federal Chancellery, whom you can see here. And just to give you some numbers, a total number of almost 43,000 people registered to participate in the hackathon. Almost 1,900 innovators gave ideas for possible projects, and almost 3,000 mentors from various fields helped the participants in developing own ideas or implementing given ones. These over 40,000 participants managed to develop almost 1,500 projects in as few as 48 hours by forming interdisciplinary teams, including specialists from IT, chemistry, social sciences, and various other fields. The resulting projects cover many important areas in the current situation. Some teams took a look into e-learning and homeschooling that already is necessary and also will be in the future, because Germany is not really prepared for doing school and university online. An important task was mental health in times of social distancing and self-isolation, and also the organization of social distancing itself. And a field that is crucial, as we can see in our everyday lives, where we almost had to fight for toilet paper in the supermarkets at some point, is the distribution of resources that is apparently problematic in many areas, but especially threatening the medical sector. Of course, most of the projects were not completely finished after 2 days, but many of the teams still continue working to finalize their ideas. The federal government also will give financial and idealistic support to some of the most promising projects. I would like to present you some of the results, not only chemistry-related ones, but also projects from other areas, with most of them focusing on locating resources and bringing them where they are needed. But as a start, I would like to present a project that shows how, despite the very serious situation we find ourselves in, a little bit of fun does no harm and can even help in educating the people. Do you know the game Plague the Dink? This is the opposite. The world is infected and you have to save it from the virus by choosing the correct measures. Possible measures are, for example, light curfews, closed borders, hand washing, but also not so helpful ones like fake news. Unplague shows in an entertaining way how diseases spread and why it is so important to take measures against the spreading, even if that means to deprive people of a bit of their freedom. This again might look rather funny with the beer, but it is about a very serious problem. The text above the beer says, "Imagine a summer without beer, help the farmers." Many German farmers rely on seasonal workers from various countries, but currently with the closed borders, they cannot come to Germany, which of course is a tragedy for the workers, but also for the farmers who fear to lose their whole harvest. At the same time, students cannot attend schools and universities anymore, and people have lost their jobs due to Corona and are desperately looking for work. Clever Ackern brings these groups together, helping farmers in saving their harvest and motivated workers to find work. Another initiative is Print for Life. It takes up an approach that has been used in various countries. As consumables like masks and needed medical equipment like ventilators are scarce, 3D printing can help to overcome shortages. I found a great initiative from Italy that uses snorkeling masks with 3D printed valves as ventilators. There are also groups that are 3D printing face shields as replacement for masks and so forth. But a problem is still to solve. There are people and organizations with 3D printers and a lot of great plans for how to print useful items, but people with 3D printers don't know which printed products are needed in their vicinity, and hospitals don't know where they can find someone with a printer to help them. And this is where Print for Life draws on. It is a platform where hospitals can order 3D printed articles they need, and everyone with a 3D printer can take on these orders and thus help the hospitals. Remedy Match also aims on helping hospitals with needed goods. Gloves, disinfectants, and masks are desperately needed to protect the hospital staff and allow for optimal care for patients. And there are still companies and individuals that have the needed consumables available and could give them to the hospitals, but no organized interchange is possible so far. Remedy Match aims on bringing offer and demand together. Hospitals can register their demand online, companies and individuals can offer their spare consumables, and volunteers take care of the transport. Sadly, Remedy Match is not online yet, but hopefully will be in the next days to help the hospitals. And now I'd like to present you a tool that is available online and has been functioning for almost a week now. It is the project I worked on in the hackathon. It's called PIRAT, which apparently is German for pirate and stands for Pandemic Important Resource Allocation Tool. In the beginning, we were 3 IT students and me, and I even asked my teammates whether they really wanted me on the team as I couldn't help them programming-wise. By now, we have grown quite a bit to a group of 19 people from very different disciplines. The idea for PIRAT was born in a coffee break with my colleagues in our institute. We knew that the university would close soon and we would have to work in home office, and we thought it would be a pity to have all our consumables and devices standing around, not being used by anyone, with us not being allowed to do lab work, while the accredited corona testing laboratories needed additional devices and workers to be able to meet the demand for corona tests. At that time, some testing laboratories were already starting to acquire skilled PhD students from universities to help with the tests. So in the hackathon, we decided to build a platform to bring together universities and testing laboratories and enable an easy exchange of resources. So on our website, universities, research institutes, and individuals can create offers online. This is done by simply entering the information about the offered resources in a form that looks like this. Of course, contact data are crucial. We check whether the entered email address belongs to a real person at an existing institution. By giving the institution's address, we can assure that resources are exchanged between institutions close to each other to avoid lengthy transports. It is possible to offer manpower in terms of students that are experienced with techniques needed for COVID-19 tests. Additionally, universities can offer to lend their devices like PCR thermocyclers or centrifuges to testing laboratories and they can also offer consumables that might be needed. If not by the testing laboratories, then maybe by other corona-related institutions like hospitals. Testing laboratories or other facilities that need some of the offered goods can search for needed resources in their vicinity. They can choose what experience and qualifications they require for staff to help them, and they can search for equipment and consumables they might need. The results then are shown sorted by postcode, and contact can be made via a contact form or directly via email. What you can see here on the left is no dummy data. It's real data of PhD students and postdocs, in this case from Munich, but we also already have offers from other cities in Germany. So as you can see, our tool is already being used. And if you would click on Show Details in one of the entries, you would see a contact form like the following one. Via this form, contact can be made between offering and requesting institution. The details of the exchange then are arranged directly between the involved parties, so that solutions can be found as fast as possible without a third party having to convey. Additionally, thanks to this procedure, special requests can be respected. And the involved parties can agree on for them suitable conditions like approximate date of return, transport, or in the case of exchange of staff, working hours, and so on. Of course, our system is expandable, for example, to goods and staff needed for hospitals or whatever else might be necessary in the future. So what we are doing at the moment is contacting universities and testing laboratories all over Germany. We already got a lot of positive feedback from potential users and also received various offers, as you can see on the left. The next step, besides smaller technical improvements on the website, will be the expansion to other countries. We have a team for Switzerland and one for the UK, but we would be happy to include as many countries as possible. Generally, the website already can be used from any country, but of course we have to check legal requirements and take care of proper advertising in every participating country to allow for as many offers in our tool as possible. If you want to check out PIRAT, just visit our website. It is available in English, and you will even find tutorial videos that explain how to offer and search resources. And if you want more information or want to spread the idea and bring PIRAT to your country, please just contact us at mail@pirat-tool.com. We are very happy to help. And before my talk comes to its end, I want to show you some of the wonderful people that form our team. No one of us could have developed PIRAT by only using his or her own skills. Everybody was and still is needed and can contribute, no matter his background. And even though many of us have never seen each other in person, we still managed to create something that will hopefully be useful. So what I personally learned from the hackathon is how great interdisciplinary teams can work if everybody is determined to fight for an important goal. And I hope that PIRAT will help at least a little bit in the worldwide fight against COVID-19. And with this, I would like to close my talk. Thank you to everyone who joined this fight in whichever way it might be. And of course, thank you all for your attention. It was an honor to speak to you.
Thank you, Julia. I think it was a really nice talk about telling us how in Germany how has the challenge been approached, like in general with different group collaboration and then collaboration in general with the public health system to show that we are all in this trouble together and that we will get all together. So I think that this is the end of the program of the presentation that we have, but we can go now for the question that people have asked and that we have more or less selected from all the different presentation and speakers. I have one for Angela and Cynthia Liu, Professor Angela and Cynthia Liu. They have a question from Lavina Kursniatai. Sorry for my pronunciation names. It's, they ask, why is it so hard to find a vaccine on the COVID-19 virus? Is it why we have to tackle so many different small molecules to find a cure?
Well, thank you for the question. I may answer a little bit, then Cynthia may have something to add on. So to develop a vaccine actually takes a long time. Not only just the designing part, it's challenging, and the most challenging part is the clinical trial, to try real person and then to test if they have antibodies developed. And that's not long enough, We also need to let those volunteers who, take the vaccine shot and just live on their normal life and compare with the control group, see, if they have less chance of getting the disease infected. So that takes a long time, at least 3 months for, phase 1. And for the phase 2, they need to test the safety and toxicities of the vaccine. And that takes another, at least 3 to 6 months. Then if they're gonna spend to phase 3, gonna, apply more people, that will take another, half year or so. So it takes, years to develop a vaccine, especially in a clinical trial phase. It's not like, the therapeutic reagents, like, repurposing the drugs that may take much less time than the vaccine. So vaccine will take longer. It's not like we're gonna have the vaccine in the next couple of weeks, no. Cynthia, do you have anything to add on to that question?
Don't know if we have Cynthia, but I mean she can connect whenever she wants and add to this conversation. There has also been a question regarding the groups of drugs that also regarded to you, Angela. About why should we focus on a group of drugs with a specific mechanism. They are asking also about should we focus on a drug that targets the virus itself rather than the cellular receptor in which it binds, and since there are differences in how much we know about both.
Yes, so not only the proteins that, virus protein, those will be good drug candidate, those will be good targets for drug. Some proteins on the human cells, like the ACE2, there's another receptor, actually, I didn't go over much, the AT2 receptor, those could also be possible drug targets. And like the paper that I think Dr. Martinez mentioned, and Professor Balogun mentioned as well in the Nature paper, they, screen in the human cells, try to figure out if there are any human proteins have possible interaction with these viral proteins. All these proteins could be possible drug targets, for us to interfere with the drug, with the viral infection process.
If I may, another reason why we are not going through the mechanism but just using known treatments is because those have been already approved, and we know that the side effects of these treatments are low. So it's not the ideal case where we are in a state of war against this virus. So we are using our current ammunition because it has been approved and we know it's not going to kill patients. In the meanwhile, for the long run, we need to develop the kind of treatment that you are suggesting when we are targeting specific points in the mechanism of this particular virus.
Yes, exactly, because right now we're really running out of time. So we wanted to, it's called repurposing of the existing drugs. Those drugs have been tested on their toxicities and the side effects on the, big population. So it's, at least it's safe to use, it's safe to use for controlled, people. It won't, it's much safer than a small molecule still in a development phase. It will take much shorter time, for those existing drugs to pass the authorities to become, available for the patients.
Thank you. If I can add one, comment on that? Yeah,
sure,
I mean,
please.
I think, of course, as we discussed, repurposing drugs is probably the fast track now, and it's a lot of advantages in terms of affordability and safety already being known. But if we think about the long term and we take let's say the experience from other infectious diseases, I mean, having different candidates with different modes of action, this would allow, of course, combinations that could avoid resistance, for example, be much more effective. If you look at HIV or malaria or tuberculosis, many fields of infectious diseases, different modes of actions combined together are usually the best tools. And if we think about the targets of the hosts, if we in the long term develop drugs that actually target the host, and this host protein, host receptor is actually shared with new emerging viruses, these could be very good tools for new pandemics coming up in the future. So it's a balance, of course, of resources at the moment. We have to go fast, but it's also important to look at different strategies to have the best toolkit to prevent the future pandemics and also treat the COVID-19 one.
Thank you.
If I may ask, can I add just a point? I think,
sure, Emmanuel.
Yes, really, at this moment, I think what will help us in solving this pandemic will be if we focus, of course, there are lots of interest, but if focusing more on the viral factors will be a very faster way to go, I think, in terms of drug development. I don't know if Angela or Javier will agree with me on this.
Sorry, I wanted to add, Antonio, can you unmute Cynthia? She says she's, muted by the host. If you can unmute her, then she can join the discussion as well.
Okay. Yep. No, it should be.
Yes. What is question? I'm sorry, Emmanuel.
No, it's not a question. It's a comment that I made that if we focus more on the viral factors, I think we will be faster at getting to solutions rather than focusing on the host factors. Just my view.
Yes, yes. I think different people may have different approaches, and we have, scientists global wise, we all focus on this matter. Some people may focus on virus, some people may focus on human side. Yeah, it's all contributing to the event.
Okay. I want to go also to Emmanuel. We have a question for you regarding the biochemistry or compatibility of the virus with the human cells. Unai Alastrue is asking, why is this virus to human cells so strongly?
Okay, I guess that question is for me.
Yeah.
Yeah. So yes, like I showed you in that data of that the work that was published in Nature, which myself and Javier showed, that there was a mutation in the SARS-CoV, that's the previous SARS coronavirus, and these mutations resulted in change of some amino acid residues where at the RB, that the receptor binding domain of the spike protein, and this amino acid change, whenever you have an amino acid change, there is possibility of conformational changes, even if there's no conformational changes, but you may have extension of R sites, that's R group, and then that R group will lengthen to increase the binding of the receptor, and that is what that data showed, which is really remarkable, and how it has been tied to the infectivity rates of the virus. So that the mutation that occurred resulted, that nucleotide change that occurred resulted in changes in amino acid, and this changed amino acid increased the binding strength or affinity to the receptor on the human cell.
Okay, thank you, Emmanuel. I just want to quickly address one issue before, because we are seeing that a drop on the people regarding the next session for tomorrow, the second session. So tools to try remotely. I wanted to ask Joao Borges from our team of organizers that he's going to be the main coordinator tomorrow. So if you wanted to go ahead and to present the next session, or Joao, are you there?
Yes. Can you hear me?
Yes.
Yes. Hi, everyone. So first of all, I'd like to thank again to all the speakers for these very nice talks and about the COVID and what is the role of chemists in this in fighting against these pandemics. So I will just go briefly into the session that we'll have tomorrow. It will be more related with the tools, as Antonio mentioned, to how to succeed while we are working from home. So this will be useful not only during this crisis, but also through life. I think that we'll experience similar circumstances that we are facing today, not only related with disease, but other issues. So as you can see, we have really a fantastic lineup of speakers. We'll be also hosting a Q&A panel discussion at the end. And of course, as you can see, we'll target different issues so that several of us are having. So how to teach class remotely, how to find useful tools online to combat this COVID pandemic. Also, how to keep our work-life balance, how to keep healthy, and how to communicate chemistry in a creative way. And of course, in the end, we'll have this panel discussion, as I mentioned, that will last for around 30 minutes. Before that, I would like just to highlight, and can I share the screen, Antonio?
Sure, sure.
Can you see? No.
I will make you.
Wait.
Now you are the presenter.
Can you see now?
Yep.
Okay, so this is, I would like just to introduce this opportunity for us. So there is also a module on promoting mental health that is being promoted by the Northeastern University, as you can see here. So please, I encourage you all to take actions and to follow up. This is quite provides nice tools on how to proceed during this crisis and how to address it, how to keep safe. So I would be pleased if you could also check it out before our webinar by tomorrow. And of course, we will be very glad to have you all by tomorrow again.
Thank you, Joao. Just to remind everyone, will be tomorrow at 3 instead at 4. It will be at 3, and you can check on the link on the social media and the GoToWebinar links that we will publish together with the program. So we can continue with the Q&A if all the different speakers are still available. If you can stay for a couple of minutes, 5-10 minutes. I have a few questions that are really interesting for all of you. So Emmanuel, there were a few questions regarding the high temperature comment that you said. Also, I think that Jadel mentioned something. Is it how true is that the virus is resistant or not resistant to high temperatures?
Yeah. Okay. Yes. From studies show that actually this virus, that is this SARS-CoV-2, at least, it's relatively level to high temperature. And I think the survival, from what I understand, is that it doesn't survive for long above 30 degrees centigrade, above 30 degrees centigrade. So, and this may be one of the underlying factor that helps to promote the infectivity. You know, as you remember, it started around the winter period in many countries, except of course for countries like ours. Most African countries are, tropical regions, so where the ambient temperature is usually much more above 30 degrees centigrade. So some people have attributed this to be one of the helpful factors for Africa, other tropical regions, as you can see around the world. So it's really labile. And again, maybe I should also use this opportunity to share with you. This has not been scientifically proven, but we know here in Africa that although the temperature is very high, but people still consume very hot foods. So, and some people locally are saying that consumption of hot foods may also be helpful, but we don't know, at least for now. So this needs to be substantiated just beyond empirical knowledge. Yes.
Okay, thank you. Jadel, you want to add something to that?
Yeah, maybe I can add them. I think my comment was more related to, let's say, seasonality and climate and temperature directly. It's still very early days, you know, coronavirus 2 or SARS-CoV-2 is not SARS-CoV or MERS or other viruses, but many other viral diseases, they show seasonality. Some peak during winter, like flu or noroviruses. Some others peak during summer, like dengue, for example. I think it's very early days for SARS-CoV-2 to see if it will become seasonal or not. There's a good chance, I think, from previous studies with other coronavirus, that it might become seasonal during the fading phase of the epidemics, but it is still very early days. I mean, just look at the distribution of the cases around the globe. I mean, it affected summer, winter, it doesn't matter. I mean, the virus is so infective that it actually didn't mean that. But there are some hints, unpublished data, unverified, that show that maybe the higher number of cases are going through more winter areas. But it is still early days, not proven, and I think that's not critical or the main component of the epidemics. I think other parts, like the infectivity of the virus, is the way it spreads, how it replicates in the upper airways, and these other components of the viral biology and the epidemics are more determinant to its dynamics than the seasonality itself. That's my view.
Okay, thank you. I think that I have a few other questions, but we have gone over the time, and I don't want to keep up with all of you with your time. I just want to remind everyone that this whole season will be available afterwards for watching online on the social media and different channels, so you can go back again to watch it and to get more information and such. So I just want to thank you, all of you, for all the speakers, all the participants, for the attendees, for joining. Special thanks to IUPAC and Chemists, International Younger Session, and all my colleagues on the European Young Chemistry Network as well. Without their help, this wouldn't be possible. And I think that maybe Professor Javier Garcia may have some final words for all of us.
Just to thank you, all of you, especially the participants. I think we really had a global conversation, perspective from all around the world, very different angles to this pandemic. Very clearly, scientists have a key role to play. I think one of the key messages of this webinar is that we need to go to the right sources of information. I think that the main purpose of these webinars is that people can get informed from experts. So please follow the advice of your authorities, go to the World Health Organization, try to be well informed, inform your colleagues. Please visit us again tomorrow at 3 p.m. Central European time. I want to congratulate the organizers. I'm so pleased, I'm saying this on behalf of IUPAC, that is the young chemist who had the idea and the leadership to put these two sessions together. For most part of the webinar, we were running at full capacity. That is, when we were talking, I never thought that we will exceed the capacity of this webinar, 500 participants, and I have to apologize to everybody who couldn't join at the beginning where we exceeded the 100 people limit. But I think that's also a clear indication of the interest of the public to be well informed. So please visit us again tomorrow, 3 p.m. Central European time. Thank you to the organizer. Thank you to the participants. And have a nice day. Please stay safe, and healthy.
Thank you.
Thank you, Javier. Thank you.
Yeah.
So thank you all, and see you tomorrow. Then you have the, as Javier was saying, the program there. Feel free to join at any time, and be early. You have seen that we get the full session.
Yeah, be early. Okay. Now,
Bye, everyone. Ciao.
Bye-bye.
Bye.
Thank you.
Thank you all. Yeah, bye. Thank you.
Okay, we